The Sustainable Development Goals are a set of 17 global objectives established by the United Nations aimed at combating the world's most significant environmental, ethical, and economic difficulties by 2030. Today we will be focusing on SDG 14, Life Below Water, specifically Target 14.3, to minimize and address the impacts of ocean acidification. This presentation will depict the biological impacts of ocean acidification on a molecular level, explore how progress is currently being tracked in regards to Target 14.3, and demonstrate how current molecular research and technologies approach this target. We will also reflect on future directions, suggesting potential initiatives to progress towards Target 14.3. Ocean acidification matters due to its disrupting the molecular equipment that marine life depend on. When the pH drops, enzymes lose their shape, slowing down the vital reactions like metabolism and growth. Cells then spend more energy balancing ions, leaving less for reproduction and repair. At the genetic level, organisms switch on stress pathways instead of normal development, weakening their resilience. These molecular changes accumulate into weaker reefs, biodiversity loss, and risks to biodiversity, ecosystem structure, and food security. That's why SDG 14.3 is so important, as protecting ocean pH means protecting the molecular foundations of marine ecosystems and as well as the society we rely on them.
The health of ecosystems in the ocean is heavily dependent on the function and stability of the biological cellular processes and molecules. Ocean acidification is the result of an increase of hydrogen ions in the seawater caused by the chemical reaction of carbon dioxide and H2O, creating carbonic acid and hydrogen as products. The hydrogen ions decrease the pH of the ocean. This reaction of ocean acidification can cause significant damage to marine ecosystems by reducing the available amount of carbonate ions, which are essential for many vital organisms that rely on calcium carbonate structures, such as corals, planktons, and mollusks. Protecting ocean pH is essential for preserving molecular functions that SDG 14 aims to protect.
In order to reduce carbon dioxide and mitigate ocean acidification, molecular biology strengthens focused conservation efforts, educated adaptive management, and offers early warning systems. Molecular biology's indicators include plankton studies to follow adjustments in metabolic pathways associated with decreased energy from acidification, biosensors to measure the availability of carbonate ion in seawater, and genetic biomarkers like oxidative stress proteins that expose early signs of coral stress. In addition, managing and reducing other pressures on ecosystems like fossil fuels is also beneficial for coping and identifying areas of vulnerability and resilience. The Marine Estate Management Authority is an exemplary approach initiated nationally to manage the New South Wales marine estate as one continuous system. Through targeting water quality improvement and delivering successful governance, New South Wales can achieve the pressure reduction on marine organisms and ecosystems. Several existing frameworks have also been established internationally to support the tackle on ocean acidification, including the Paris Agreement 2015 to cut CO2 emissions, UN Decade of Ocean Science, which promotes international collaboration, research, and data sharing on ocean chemistry, and the Convention on Biological Diversity to encourage countries to integrate acidification into biodiversity planning.
Ocean acidification does not solely affect the Earth's chemistry, but its biology too. A lower pH and increased concentration of carbon dioxide can severely impact organism survival, not only through hindering calcification required by numerous marine animals, but also through its interference with basic molecular processes, these being unfavorable conditions for enzymes. Acidic pH levels increase hydrogen ions. These extra ions interact with the protein's amino acid side chain, changing their charges and disrupting peptide bonds. If this disruption is drastic enough, the final shape of the enzyme will be altered. Distortion at the active site will denature the enzyme, meaning that they can't catalyze reactions, causing cellular dysfunction. Disrupting cell membranes and transport. The pH change disrupts the ion equilibrium across membrane walls, affecting the ability of cell membrane pumps to maintain homeostasis as ATP is required. This extra energy expenditure on ionic homeostasis impairs the cell's ability to perform regular functions. Cell signaling. The drop in pH and hydrogen ion increase can activate stress response pathways within the cell, interfering with the movement of calcium across membranes, disrupting cell signals. This diverges necessary energy from cellular functions to stress survival. 3. Gene expression. The extra carbon dioxide from the ocean diffuses into the cells of the organisms, causing metabolic depression. This allows for the organism to conserve maximum energy whilst maintaining ionic homeostasis. This is then reflected in gene expression.
CRISPR-Cas9 is a precise gene editing tool that could help address ocean acidification by enhancing the resilience of marine organisms. Acidification reduces carbonate ions, weakening the shells and skeletons of species like corals and shellfish. CRISPR offers solutions by targeting genes involved in calcification, acid-base balance, and stress tolerance. In corals, this could sustain reef growth, while in shellfish, it may support aquaculture and food security. Future research is exploring multi-gene edits and aquaculture trials to test safety and effectiveness. However, delivering CRISPR into wild populations is a major barrier, and strong international regulation will be needed to ensure the technology is applied ethically and sustainably.